Our unique drug delivery system, which uses 20 nanometer gold nanoparticles in two specific short peptides, can stabilize gold nanoparticles in physiological solution and enhance its cellular uptake, and thus effectively deliver peptide-based or other drugs into cells. Our GNP peptide hybrid is easy to synthesize and stable. By using gold nanoparticle peptide hybrid as drug carriers, we can transfer peptide drugs into the cells effectively and in abundance. We treated ischemia-reperfusion injury in the lungs with our GNP-PKCI hybrid. GNP brings PKC inhibitor peptide into the cytoplasm. This delivery method was able to reduce more severe lung injury using a lower dosage of PKCI than other delivery methods.
To begin this procedure, retrieve the peptides from a freezer at -20 degrees Celsius and thaw them at room temperature. Use a microscale to weigh 0.01 gram of each thawed peptide and transfer each peptide into a separate 50 milliliter conical tube. Add 18.74 milliliters of deionized water to the P2 tube and 16.93 milliliters of deionized water to the P4 tube. Add 8.21 milliliters of 50% acetonitrile diluted in deionized water to the PKC delta inhibitor tube. Vortex the peptide solutions briefly, then place the tubes into a sonicator at 40 MHz for 5 minutes. After this, transfer the solutions to a